Good morning, good afternoon, or good evening, depending on where in the world you're watching from. Thank you for joining us for the fourth day of Cell and Gene Therapy Digital Week, a live webinar series brought to you by the producers of the face-to-face conference, Cell and Gene Therapy, Bioprocessing, and Commercialization. This year's event takes place in Boston, September 25th to 28th as part of Biotech Week Boston. I'm Eric Warner, Marketing Manager at Connect 365, the world's leading facilitator of knowledge sharing and business connections. We hope you enjoy all the expert-led sessions and resources available to you as a Digital Week attendee. If you like what you see, please click the Invite a Colleague button or click one of the on-screen social media buttons to invite others to register for free. This session is titled Building a Sustainable Academic Edge for Feeding De-risked Assets into the Biopharma Pipeline. And it's being presented today by Dr. David DiGiusto, Executive Director, Stem Cells and Cellular Therapeutics Operations, Stanford School of Medicine. Should you have any questions during the presentation, please type them into the control panel below the slides, and we'll do our best to answer them during the Q&A session immediately following the presentation. Without further ado, I'll turn it over to Dr. DiGiusto.
Welcome, and thank you to the organizers for the invitation to discuss our development of cell therapeutics at Stanford University. My name is David DiGiusto, and I'm the Executive Director of Stem Cells and Cell Therapeutic Operations at Stanford. And today I'll be discussing the risks of developing academic cell therapy products and how we mitigate those risks. On the current slide, you can see that I've listed some of the issues that represent risks in the development of cell therapy products. They include the pipeline, production systems, and lack of clinical infrastructure. So in our pipeline, we often have too many projects. They won't all succeed. They come from lab protocols or not standard operating procedures. We haven't really assessed or developed in-process metrics prior to development, and many of these products are underdeveloped or not qualified in terms of the product release assays. With respect to the production systems, some of these systems are non-scalable or open, so they're not amenable to clinical implementation. There's often a high cost of goods associated with the production system. There may be insufficient raw material specifications or sole source providers which limit the availability of materials. And sometimes reagents are of ruminant animal origin, so they have insufficient certificates of analysis or no certificate of origin preventing us from using these materials. In many cases, there's a lack of automation, which means we introduce operator variability, and that can lead to variability in the resulting product. Additionally, with respect to clinical infrastructure, we often have inexperienced physicians and nursing staff and or no clinical regulatory affairs to govern the trial, and we often have a lack of clinical space dedicated to translational studies. In an effort to address these issues, we have created what we call the Stanford Center for Definitive and Curative Medicine, and we hope to be able to use this center to address some of these issues and bring our cell and gene therapy products forward. So what do we hope to achieve with the Center for Definitive and Curative Medicine? These slides list some of our goals for the center, including pipeline development, product selection, development of a business model, and attracting commercial products at the end of proof of concept studies to aid in the commercialization of products. Some of the key infrastructure enablers for the CDCM include a fully integrated team with expertise in stem cell biology, gene therapy, immunology, hematology, and oncology, the development of manufacturing capabilities with the capacity to prepare customized stem cell and gene therapy products, infrastructure for proof of concept clinical trials with regulatory and development expertise for therapeutic cellular products to treat patients with genetic and complex disease, and a sustainable business model that will drive innovation and translation of ideas to the clinic and beyond. And in fact, we hope to use this to be able to keep the pipeline alive and continue to bring new products forward as products exit the pipeline. I would like to recognize and acknowledge some of the key players in the Center for Definitive and Curative Medicine. I include my colleagues Tony Oro, who is a professor of dermatology, Dr. Maria Grazia Roncarolo, who's a professor of pediatrics and the chief of our Division of Pediatric Transplantation and Regenerative Medicine, Dr. Matt Porteous, who's the Associate Professor of Pediatrics, Dr. Judy Cesuro, who's a Professor of Medicine in our Bone Marrow Transplant Department. I'm listed there. I'm responsible for the Laboratory for Cell and Gene Medicine, which is our production facility, and my colleague Crystal Mackel, who's a Professor of Pediatrics in the Associate Director of the Stanford Cancer Institute. And I'll be featuring the work of these folks as we move through this presentation. So one thing we're doing is very much leveraging Stanford's long history of expertise and achievement in medicine, and especially in the area of cell and gene therapy, to ultimately benefit our patients with incurable disease. And as you can see here, we have a large number of, in the School of Medicine of members of the National Academy of Science, 42 members of the Institute of Medicine, 4 MacArthur Geniuses, 19 Howard Hughes investigators, and 17 NIH Innovator and Young Innovator Award recipients. And these are the type of faculty that are bringing forward cell and gene therapy candidates for further consideration. The picture on the right just kind of goes to our history of 116 years of innovation. And we see Norman Shumway here discussing a transplant, the first heart transplant into a 54-year-old steelworker, Mike Kasparek, who survived for 14 days. This is back in 1968. And it goes to Stanford's long history of innovation in the field. In addition to our faculty and their achievements, we have quite a few areas in Stanford medicine that are represented on this slide with a large number of opportunities. We've also had to develop a way to filter through potential projects and prioritize in our queue. So currently we're leveraging our programs in metabolic diseases, in tissue-specific genetic diseases, in cancer and blood disorders, and a new center for genetic immune diseases as areas of clinical investigation that we'll be approaching. This includes cutting-edge science, innovative diagnostics, state-of-the-art technology, and over 30 projects from these areas coming into our queue. And so one of the first, again, the first steps is to curate the list of projects to de-risk the assets in these projects. The next slide here shows our project review and prioritization strategy. This begins with translational staging, in which we assess the translational readiness of each project through a review of the overall project objectives, regulatory, processing source materials, and financial status. As you can see on the left here, points are given for the level of readiness as shown in the diagram. And if a project receives at least 6 points overall for its project readiness, it's ready to enter the development queue and is prioritized by a team of Stanford leaders, including representatives from our School of Medicine Dean's Office and executive staff from each of the 2 Stanford hospitals, the adult and pediatric hospitals. We use this algorithm to review all our projects and assign them a priority for entering our queue for further development. In the next slide, we're going to start discussing some of the specifics projects we have. And in order to acquaint you with some of the top projects, I'd like to first discuss the fact that CAR T-cell therapy is moving rapidly towards commercialization, and we are working to improve outcomes by developing CAR T-cell therapies at Stanford. You can see here from these Nature, Science, and Cancer Immunotherapy journals how prominent CAR T-cells are in the field. And eventually it's hit all the way through, common non-scientific magazine. Time gave it one of the breakthrough of the year in 2016. So these are very important therapies, and my colleague Crystal Mackel is developing a CD19, CD22 bispecific chimeric antigen receptor to prevent escape mutants that lose CD19 in refractory B-cell lymphoblastic leukemia, diffuse large B-cell lymphoma, mantle cell lymphoma, and chronic lymphocytic leukemia. I'm not going to spend too much time on this because I think the subject area is well publicized and well known to you all, so I'm going to move on to some other candidate therapeutics we have in the laboratory. One of the first candidate therapeutics is the administration of a monoclonal antibody to create space in the marrow for stem cell transplantation without the toxicity of chemotherapy. So we are exploring the use of a non-chemo, non-radiation conditioning of patients to prepare them for hematopoietic stem cell transplantation. This change in approach would revolutionize stem cell transplantation, opening up the procedure to sensitive patients, very young and very old, as well as patients without cancer, for example, with monogenic diseases. And we enrolled the first patient on this trial in October 2016, and we now have a second patient on the trial. The idea is to demonstrate that we can create an environment to transplant hematopoietic stem cells without the toxicities of chemotherapy or radiation, so that we have better outcomes in our patients without the side effects of these preparative regimens. Another candidate therapeutic that we are developing include the creation of regulatory T cells for suppressing autoimmune responses that result in severe disease states. And my colleague, Dr. Maria Grazia Roncarolo, has pioneered this technology of transferring FOXP3 gene into CD4 positive T cells to treat several indications, including IPEX disease. And the patient cells are taken, CD4 cells are purified, and then gene modified with a FOXP3 gene that is deficient in these patients with IPEX syndrome. And we are now developing these therapies in our laboratory through preclinical validation of this process, where we optimize the existing vectors, we're characterizing the cells in vitro, and we're establishing the in vivo function of these cells, these gene-modified cells, and doing in vivo safety assessments, while at the same time developing a manufacturing process that is compliant with good manufacturing practices, including production of the plasmids, production of master cell banks and viral vectors, optimizing the genetic modification of the cells, developing appropriate assays, and performing process qualification. Again, we'll be using these to treat IPEX syndrome, which is an immunodysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. Again, because there's no regulatory T cells in this syndrome, there's uncontrolled autoimmunity. And so we hope to develop this to prevent the severe sequelae that occur from this disease in vivo in these patients. Another area of investigation is into epidermolysis bullosa, and this is a disease caused by a deficiency in collagen 7 that leads to severe skin blistering and infection. And this disease is devastating in most children who die before the age of 7 without treatment due to infection. Our process includes the isolation of keratinocytes from the patient, followed by viral transfer with a retroviral vector of the collagen gene that's missing in these patient cells, and then creating an epidermal sheet from keratinocytes that have been gene modified. That epidermal sheet is then grafted onto the patient, and the patients then are protected in the areas where the grafts have been transferred from infection, and we typically are doing pressure points such as the hands, the hips, the knees, the elbows, where patients most often come in contact with surfaces. 4 patients have been treated with 6 wounds each. There's been, I think we're now up to a 22-month follow-up, and we've seen good safety and efficacy, and this has recently been published. There are some limitations to this therapy, however, and that includes one of the difficulties is having enough material to create enough grafts to cover the body. We often can make 6 grafts, but it's insufficient to cover the whole body, and that it takes a long time to make the product, and there are some potential safety concerns of a gene-modified cell product that varies from patient to patient. And then there's potential for immunological and biological side effects of the continual overexpression of the defective genes. To address this, my colleagues, Dr. Tony Oro and Marius Vernig, are working in their laboratories to take these same skin biopsies, but to reprogram these skin biopsies into induced pluripotent stem cells, which can be propagated in vitro or in tissue culture, where we can genetically correct the disease and have a source of unlimited source of cells from which to make skin grafts. And we can also screen the skin grafts or the cells that we make for safety, discarding the cells that might be deleterious based on vector insertion or their differentiated properties, and then just provide in vitro expanded and differentiated human skin grafts in unlimited quantities to improve the transplantation of the patients. And since most of this work can be done upfront and very quickly, we could potentially create a bank of cells that could be used for more than the patient from which they were derived. This is work ongoing in the laboratory and subject of upcoming clinical investigation. Another area of investigation in our lab is the treatment of sickle cell disease. And to do this, we are using a genome editing approach, which involves using a CRISPR-Cas9 protein and RNA guides to specify the target for disease, which is the hemoglobin locus. And basically what we do is we use the CRISPRs to create a cut in the hemoglobin locus at the site of the sickle cell gene mutation. It's a single point mutation. And once that cut is made, we also deliver a donor sequence with the wild type sequence for hemoglobin using a recombinant AAV6 donor vector. And this results in targeted integration of a wild type sequence for hemoglobin as opposed to the sickle cell hemoglobin. And we've shown that using mRNA to encode the CRISPR-Cas9 is not as effective as using a pre-established ribonucleoprotein complex. So in figure panel B here, you can see that cutting is on the average more frequent using a ribonucleoprotein. That's the RNP column versus mRNA. And we've also shown that using the AAV vector and using a GFP marker in that AAV vector, we can show that we get very high levels of integration of a AAV donor template. Again, we're using GFP to demonstrate the level of integration, but ultimately this would be correction of the mutant gene with the wild type gene. And you can see here we had up to 22% integration in this slide of the corrected gene product. But when we actually sort out those corrected gene products and we do a transplant into mice of the corrected blood stem cells, in the first recipient, we can see that we get very high frequency of gene-modified cells, and that those cells, when transplanted into a second mouse recipient, maintain the gene modification, which means we are, in fact, modifying a very primitive hematopoietic stem cell, in this case, with durable reconstitution activity. So the potential for long-time genetic modification and correction of this disease is very high based on the evidence from this model. In terms of efficacy, we know the starting DNA is derived from CD34 positive hematopoietic stem and progenitor cells, which contain 100% of the sickle gene, their defect. After we edit the DNA, we see that we have 55% of the alleles corrected, 35% are mutated, and 15% remain unchanged. When we look at the actual protein circulating in mice transplanted with these cells, we can see that we are making 86% corrected hemoglobin, and in sickle cell disease patient, if the level of the correct adult protein is greater than 70%, the patient is cured, and this is known from transfusion reactions. So we believe we're in the right range of being able to make a product that results in enough hemoglobin to cure a patient with sickle cell disease. Now I'd like to turn my attention to the development of infrastructure to help us develop and commercialize cell and gene therapy products. And this is the creation of Stanford's new Laboratory for Cell and Gene Medicine. The Laboratory for Cell and Gene Medicine is a multi-product biologics production facility. This picture here shows you some of the complex air handling equipment we need to maintain an appropriate environment. And on this next slide, I list that the LCGM is a CGMP, or Good Manufacturing Practice Compliant Manufacturing Facility. We have about 7,500 square feet of classified clean rooms. That includes 4 cell culture suites, 2 viral vector production suites, a fill and finish suite, a media preparation room, release materials room, and an irradiator for making irradiated feeders. We have another 1,100 square feet of controlled support space, including dry storage, gas storage, raw materials and product storage, biohazardous waste, and USP purified water. We actually use the USP purified water to clean the facility. We have another 150 square feet of quality control laboratory where we perform molecular and cellular analytics for our products and reagents. We do compendial testing such as sterility, mycoplasma, and endotoxin to ensure the safety of the product. And we do all our environmental monitoring assessment in the QC laboratory. Additionally, we have 10,000 square feet of process development space, and this is where we develop the products that come to us from a research laboratory into products that can be manufactured and used in the clinic. This includes cell processing devices, bioreactors, chromatography equipment, standard tissue and viral vector and bacterial culture capabilities. All the equipment we have in our GMP facility is reproduced in our process development lab, so procedures created or developed in the process development space can readily transfer into our clean rooms for ultimate clinical manufacturing. Next slide is an example of what it looks like in our cell processing clean rooms. You'll notice not much clutter, just our biosafety cabinets, incubators. On the ceiling, you notice HEPA filters for filtering the air to ISO 7 cleanliness. The floors are poured of a single epoxy coat of flooring, so there are no seams, no cracks, nowhere for dirt or dust to get trapped. The rooms are monitored routinely and cleaned and maintained by professional cleaning services and are turned over every time we make a different product. The room is completely cleaned and monitored environmentally for particle counts and microbacterial load to make sure that we are compliant with federal regulations for clean rooms. This is another example of our clean rooms, and you can see on the left here the gowning ante area where we, before we go into a clean room, we put on all our clean room gowning and ensure that products that are brought in there are all in appropriate packaging. No cardboard, no paper goes into these rooms. And on the right, you can see one of our clean rooms that has in the biosafety cabinets on the left 2 influx cell sorters for isolating hematopoietic stem cells and regulatory T cells that are contained within biosafety cabinets to ensure in this process we maintain the sterility of the products and protection of the operators. This slide, the next slide gives you an example of our staffing. Our staffing is broken into folks for regulatory affairs to help develop investigation new drug applications, specifically the CMC sections for products that are brought to us. We have operations people that manage the facility, take care of administrative paperwork, business agreements with our customers and our vendors, a process development and manufacturing team that both develop these processes but also manufacture the products once they've been developed, and then a quality systems team that governs our compliance with federal regulations, state regulations for manufacturing cell products, controls all our documents, raw materials, final product release. So this is very typical of a academic manufacturing facility team to shepherd products forward. So I want to be clear that it's not just the manufacturing component, but all the regulatory quality system and operation components that make this possible at our institution. The next slide is really sort of a diagram of the activities we perform to de-risk cell and gene therapy products through careful process development and clinical trials. Starting over at the left, our process, as I mentioned earlier, starts with a project review. That review is based on scientific, technical, regulatory, clinical, and financial aspects that I already talked about. Once a project is reviewed and brought into the facility, we'll start out with process development, which includes reagent qualification, either scale up or scale out, where scale up is making more of a product and scale out is making more products for more patients. We perform assay development to characterize not only the raw materials, but in-process and final products are characterized through assays that we perform at our laboratory. And that allows us to establish product specifications for both in-process production and product release so that we can have metrics by which we measure the quality of our products. Once we've developed all the processes and we've documented those in standard operating procedures, we perform process qualification to ensure the robustness of the process, and that includes at least 3 engineering runs where we make a full-scale product. We perform full release testing. We will use those materials created in our process qualification to support toxicology and stability studies, and we will write the chemistry, manufacturing, control section for the investigator's investigation of new drug application in support of that regulatory requirement. Once we move on then to clinical manufacturing, we'll do all the raw material staging, the production of the product, testing of the product, release and archiving of samples of the product for follow-up, and then we will move to support of clinical assessment once that product is administered, including the safety, the feasibility of manufacturing and delivery, the efficacy of the product, and the indication for which the product best works. We do this by supporting the clinical investigation with information from manufacturing. And then finally, we also perform correlative studies on the pharmacology, toxicology of the product, any immunology related to product acceptance or product activity. In many of our gene modification and or lentiviral vector transduction projects, we perform genotoxicity analysis, and we work out a mechanism of action where possible for the products. Now, many times through these clinical assessments and correlative studies, we either receive new information or have less than optimal outcome, and we go back to process development to sort of work out the kinks in the product, to then take it again through the manufacturing and clinical assessment. This is an iterative process for most products until we get the product just right. So the point here is that this is not a simple one-through service provision. This is a collaborative effort with our investigators to help develop the products in a great level of detail. By developing the production and the regulatory strategies, we are de-risking the products. So when we identify potential commercial partners after proof of concept, we have taken a lot of the risk out of the product during process and product development. And this is all supported by our regulatory affairs and quality systems infrastructure. Another way we take some of the risk out of the product is by using closed systems, scalable technologies, reagents, and automation. And on this slide, you can see some of our closed culture devices and plasticware. On the left, you see a gas-permeable bag that we use to culture cells as opposed to tissue culture flasks or Petri dishes that are often used in the research lab. This allows us aseptic transfer of cells and media into and out of these bags for culture, thus reducing the possibility of contaminants. Similarly, we've modified common laboratory devices such as centrifuge tubes, where we've put in weldable tubing so the transfer of materials that need centrifugation is done through a closed weldable system. So we, again, reduce the likelihood of contaminations by using closed systems. Just below those figures under automation, you can see here one of our cell washing devices, the Fresenius Lovo. And in this case, the device is used to wash products free of platelets, and in some cases following red blood cell lysis of the hemolyzate. We're able to wash, formulate, concentrate cell products all in a closed system with sterile tubing welds, and that allows us to do, in a very safe manner, the upfront preparation of processes for downstream activities such as cell separation and cell culture. To the right is a closed system cell separation device. This is the Clinimax Prodigy, and we're using this to manufacture our CAR T cells, which includes upfront washing, magnetic bead labeling, cell separation to enrich for CD4, CD8 T cells, the addition of viral vectors to introduce our CAR constructs, and then the final washing and formulation of cell products are all done on this automated device to take technician error out of the equation and to ensure the reproducibility of our processes. So this, again, goes a long way towards de-risking assets if we have an automated, reproducible process. Then to the right, where we have a disposable, scalable bioreactor. Many times in the academic laboratory, viral vectors are grown in cell factories or cell stacks, and that is not very scalable, so we are working currently on scaling up viral vector production to be performed in perfusion bioreactors so that we can very much scale these processes to meet demands. And for commercial demands, our pilot scale perfusion bioreactors are then scalable to commercial production scale bioreactors. So we're working out these processes with our vectors now, so as we transfer those to commercialization, they're readily scaled. And then we use clean reagents, and there have been some issues with trypsin products that are derived from porcine sources, and so where we need to do cell dissociation, we've begun using reagents that don't come from mammalian sources, so we eliminate the possible presence of mammalian viruses. And these products are currently, we're working in collaboration with the manufacturers to develop this as a GMP product. So these are all examples of ways we address risk in product development and product scaling as a way to improve the quality of our products, de-risk the assets, so when we go to commercial partners, there's a much more attractive product for them to in-license. I'd like to turn now to establishing the clinical paradigm. Once you've made all these products, you need to deliver them to the patient, and that has a whole set of infrastructure that supports that. You can see on the right here my clinical laboratory staff who are responsible for the delivery of the products we make to the actual patients. They ensure the product quality, the product prep, and that the right product goes to the right patient. I can't emphasize enough how important that activity is in order to have a sustainable, successful clinical trials infrastructure. On the next slide, I'm diagram here sort of the investment in the delivery channel. And there's a life cycle to our definitive and curative medicine where there's 3 distinct phases where we've already made investments, and they are basic research and product development, and then also clinical research. And we start out with, having an idea. We validate the target or the approach. Then we perform preclinical development, followed by clinical product manufacturing that I just went through, and then clinical development along with the phase 1 and phase 2 clinical trials. Now, at each of these stages, there are certain requirements. I've gone through the upfront requirements, but in the clinical stage now, we really need to develop infrastructure for regulatory and compliance activities. So there are key personnel to manage the IND process, the investigation of drug application process, and regulatory compliance. And then we've created data safety and monitoring boards to oversee the clinical research information. And then we are starting to develop the technology to support our clinical data management. So we have some small to medium scale clinical data management solutions we're working on so we can keep track of all the clinical data. We have invested some capital in unique clinical resources, including dedicated beds in our hospital for investigational studies, and they need to meet the standards for investigational trials in cell gene therapy. So this is not just a standard of care unit. This is a very special unit with high levels of monitoring of the patients. And then probably most importantly is, we've invested in people for these trials, including a dedicated medical home that aligns the clinical research outreach and the administrative staff. It's a nucleus of ancillary and support services and additional clinical investigators to manage the increased patient volume. So this slide demonstrates the investment at the clinical research scale to ensure the likelihood of success. Now, in addition to that investment, the Lucile Packard Children's Hospital is building a new 5-story hospital in which we have, again, dedicated space for our pediatric clinical trials. And in the same vein, the new Stanford Adult Health Care Hospital that is going up has, of course, also incorporated clinical trial space to support the evaluation of trials that are targeting adults. So these are 2 major investments of Stanford Medicine to support the translation of these trials to the clinic and careful clinical evaluation through phase 2 proof of concept. Again, I can't emphasize enough how important this is to our success. So another step in this process is to leverage our existing infrastructure, and typically, or ideally, this is our blood and marrow transplantation cell therapy facility. So, of course, bone marrow transplantation has been the hallmark cell therapy that's been ongoing for 30 years now, and our standard of care services deliver over 400 products per year. I think actually we're now, since making this slide, we're up closer to 500 products per year, and that includes autologous and allogeneic blood and marrow products, cord blood products, and donor leukocyte infusions. We distinguish, they make public health service designated 361 clinical materials. So for minimally manipulated products, they deliver these products to the clinic. We have a cell pharmacy for our corporate collaborations. We have a number of trials in which we are a collection site and an administration site for cell products that are manufactured elsewhere. And so they are collected, shipped, received, thawed, and served to our patients. And that's an entire operation within the BMT unit. And we are, of course, accredited by FACT, the American Association of Blood Banks, and FDA licensed to do this. So we're leveraging this infrastructure to achieve our cell therapy productions. This is essentially our clinical interface between the clinic and the manufacturing facility is the blood and marrow transplantation cell therapy facility. This is an outline of the floor plan for the transplantation cell therapy facility. As you can see on the top, there's an investigational cell processing and cell pharmacy. Again, these are for the 361 products. We have some office space, a QC laboratory, workrooms, and then our standard of care cell processing space for those 500 products that we deliver every year as part of standard of care. So this is space that we're utilizing in the hospital to translate some of this work from the bench to the clinic. And one more important point is, academic and business modeling and sustainability. Who's going to pay for all this? Where does the money come from? And you can see there are many sources of money here, ranked from financial risk to timeline to achieve value, including money we get from direct patient care, industry-sponsored trials, research awards and grants, industry acquisition of data, licensing and or spin-off companies all have a role, and some things are high financial risk but very quick. Other things are not so quick and have less financial risk, and some things are both take a long time and have a high financial risk. So we really try and map this out for how we're going to pay for it. This slide shows how we identify stakeholders and define the development dynamics for these processes. And so at the very bottom of this pyramid is discovery research and preclinical development. And that's largely the activities of Stanford University School of Medicine, where the discoveries are made and we begin to develop the products. As we move more into clinical development in phase 1-2 first-in-human trials, we engage more of the translational research infrastructure, such as process development and current good manufacturing practices manufacturing. But it now becomes more of the business of Stanford Hospital and Lucile Packard Children's Hospital, because we're conducting clinical trials, and that's where most of the activity takes place. And ultimately, as we move towards commercialization and registration, we rely on commercial partners to help us make the transition from phase 2 proof of concept to phase 3 registration trials and our biological license applications with the FDA. Ultimately, we hope that at the end of the day, these cell and gene therapies turn into standard of care and become the first-line treatment for unmet medical need. On the next slide, we have recognized that funding and payer mix changes throughout cell therapy development. As you can see from this diagram here, during the initial phases of discovery, most of our money is coming from grants, foundations, philanthropy, and institutional money, whereas we move into preclinical. Now the payer mix shifts a little bit, starts to include biotech and pharma partners. That involvement goes up during clinical and phase 1-2. Commercialization, you see the academic component going down and the hospital component going down. And then ultimately for commercialization, we'll rely highly on biotech and pharma licensing agreements to get these out into the commercial space for implementation as standard of care. So there is a dynamic there in terms of where we find money and who actually pays for these activities, and we very carefully work with our development groups to try and identify this ever-changing mix of payers throughout the development of a cell therapy product. We also noted that the infrastructure uses changes during product development, and so in different disease-specific programs such as discovery, preclinical development, clinical development, and phase 1-2 clinical trials, the lighter areas are not used much during these phases, and the darker areas are used more heavily. And as you can see that early on in discovery, we're using scientific cores and process development and some regulatory affairs, and as we proceed to preclinical development, the usage of those goes up. It includes clinical trials office of regulatory affairs, and then during clinical development, all aspects of the Center for Definitive Curative Medicine and Laboratory for Cell and Gene Medicine infrastructure get implemented during clinical development. And as we go to the phase 1-2 clinical trial, the scientific cores and process development activities sort of dwindle a little bit, and we're more focused on regulatory affairs and clinical trial operations. So we're both recognizing the change in payer status, but also the change in the demand on infrastructures, hoping to recognize that as things move through the pipeline, we will have to balance the access to these facilities and infrastructure to meet our pipeline demands. while we build sustainability, we also want to avoid conflicts of interest. So we have, investigators who have spent years of their life developing these products, and we go out and we look for institutional money or foundation money or philanthropy, and we want that money to come in, but we don't. We need to have a firewall to protect each side from conflict of interest issues. We don't want investigators to even be perceived as having a conflict of interest when they make decisions on clinical trials. So we've essentially created a CDCM Business and Financial Development Office in which we intend to develop an endowment which provides financing, and the Business and Financial Development Office will determine the funds flow into the CDCM scientific clinical development, where we'll build value that then again brings funding back into the CDCM business and financial side, and all the while having a firewall to protect the investigators not knowing the source of money, not having any sort of ability to adversely impact decision making, but maintaining unbiased approach to outcomes in clinical trials, unfettered by the pressures of the finance that's used to support these trials. So I'm going to conclude there, and thank you very much for your attention, and would like to happily answer any questions you may have now via our web interface. Thank you very much.
Excellent presentation, Dr. DiGiusto. So I think we have a few questions from the audience.
Okay. So I'll start out with, where does the funding come from to get all this started? And we do enjoy a mix of funding sources, including federal and state grants, funding through philanthropic partners who have invested in our mission at Stanford, and support from our hospitals, the Stanford Health Care Adult Hospital and the Lucile Packard Children's Hospital are currently financing operations in the Laboratory for Cell and Gene Medicine, in as much as they are the ultimate benefactors of the technologies we develop, which they offer to patients through the hospital. So that's how we are funding this right now. Eventually, we hope to have out-licensed IP, which will bring in revenues from both agreements and revenues from sales. Another question we have here is, where do we find qualified employees to fill positions at our institution? This is actually a bit of a challenge. We are an academic institution, and certainly in the Bay Area there's quite a bit of industry, and we are competing with industry for staff, but we do also bring people in out of the biotech or biopharma industry to work in our manufacturing facility. We bring some people out of academic centers who have specific subject matter expertise that we hire. We certainly draw on our standard of care nursing and physician staff from the hospitals, and do training and create experts in clinical trials from our standard of care staff. And then sometimes we are very fortunate through our membership in various societies to be able to identify potential employees through just interactions with our society. So there's a number of different ways we can find qualified employees to fill these positions, but it is a challenge. A third question I have here is, how do I mitigate the effect of sole source providers for reagents? That certainly is a challenge in the industry for everyone. Some things, there may be multiple providers, but in certain cases, certain compounds, cytokines, specialized reagents, devices do come from a single source provider. So what we try to do is look at the business of the provider, make sure they have a stable, sustainable business, and we use provider audits to determine how likely they are to be in business for the next 5-10 years. Although I can tell you from my experience, sometimes the biggest companies are the ones that decide to eliminate product lines. Many of us have lived through that. But we also try to qualify multiple providers where they exist. Example I would cite is in our use of cell cytokines. It appears that while 5 to 8 years ago we might have 1 or 2 providers with fully GMP compliant cytokines, we're now seeing more and more providers bringing their manufacturing or research rates up to a level of GMP compliance. So we're able to qualify multiple vendors and then show that the reagents we buy from those vendors are equivalent in performance. This requires time in the laboratory and well-developed assays to measure the equivalence of various products. And we do a lot of interactions with our vendors in terms of kind of laying out our requirements for reagents and where we see gaps. I think we find that the vendors are very responsive to needs in the field. They certainly want to fill the gaps. So we spend a good amount of time interacting with vendors to ensure they provide us with what we really need and the quality we need. That's how we deal with sole source providers. So those are the list of questions that we currently had. If anyone else has another question, you can just populate it into the web page, and I'd be happy to answer those, or if any come in a little later, I'd be happy to respond by email if you include your email address.
All right. Well, thank you very much, Dr. DiGiusto, and thank you to the audience for watching. I think that's all the time we have for today. But don't go anywhere. The next session begins at 2 p.m. Eastern. In the meantime, you can access white papers and videos by clicking the Resources tab or watch one of the on-demand sessions presented earlier this week. Thank you very much.
Thank you.